Hello, my name is Sydney Yoho. I will be a senior at Marlington High School this fall, and I was in the science internship program. My presentation is how leucine regulates protein synthesis in skeletal muscle in a time and concentration dependent manner. Skeletal muscle and its importance. Skeletal muscle makes up 43 to 50% of whole body protein. It is also an essential metabolic active organ. Skeletal muscle serves as a source of protein to provide amino acids for the metabolic process. Skeletal muscle and cirrhosis of the liver. Skeletal muscle loss in cirrhosis patients is also referred to as sarcopenia. On average, patients with cirrhosis of the liver lose 20 to 60% of whole body skeletal muscle. How skeletal muscle works. Skeletal muscle mass is in a constant balance between protein synthesis and muscle degradation. When the rate of protein synthesis is higher than degradation, muscle mass increases. When the rate of degradation is higher than protein synthesis, muscle mass is lost. In a patient without cirrhosis, the rate of protein synthesis and muscle degradation are the same, keeping their skeletal muscle balanced. However, in cirrhosis patients, protein synthesis does not occur as quickly as degradation, and skeletal muscle is lost. This graph depicts the process of protein synthesis in skeletal muscle. As you can see, ammonia increases myostatin, which then activates the ALK5. Those then induce nuclear translocation. AGF1 activates PI3K, which activates AKT, which then leads to TSC. For those that induce mTOR. From there, leucine increases the incubation of mTOR. mTOR activates 4-BP and P70S6K. Along with this, a number of different possible mechanisms acting on initiation or elongation steps for mRON translation promote protein synthesis. How to fix the imbalance. To correct the imbalance of degradation and protein synthesis, it is believed that L-leucine directly affects protein synthesis through the mammalian target of rapamycin, also known as the mTOR pathway. Our hypothesis was that L-leucine increases the mTOR phosphorylation and consequently its downstream targets in a concentration and time-dependent manner. We believe that if L-leucine was increased in the mTOR pathway, then protein synthesis would increase and fix the protein synthesis and degradation difference. Our specific aims. The signaling response of C2-C12 mouse skeletal muscle cells were exposed to leucine were studied at 30 minutes and 1 hour in response to 3 concentrations of the amino acid compared to a control group of the cells. Methodology. The experiments were conducted as followed. C2-C12 skeletal mouse tissue cells were grown in DMEM 10% fetal bone serum. The medium was then changed to DMEM 2% horse serum. After 48 hours, the cells were then treated with different concentrations of leucines, 0 millimoles, known as the control, 1 millimole, 2 millimoles, and 5 millimoles. They were exposed in 2 groups of 30 minutes and 60 minutes. After the exposure, cells were collected and washed in DPBS. The cells were collected and protein was harvested using 500 millimoles REPA-1X buffer, 5 millimoles of phosphatase inhibitor 100X, and 5 millimoles protease inhibitor 100X. For further methodology, BCA assays were conducted on the protein. Protein were then denatured using lamellia buffer and run through electrophoresis gel and 5 to 20% triglycerine gel. Proteins were electrotransferred to PVDF membranes overnight. Immunobots were then exposed to the primary and secondary antibodies. Results were quantified and expressed as fold changes compared to controls, and all experiments were done in triplicates. The results. After the Western blots were developed, the intensity of each band was measured and led to the following results. This means that L-leucine did not alter the expression of myostatin or IGF-1 in murine myoblasts compared to controls. Phosphorylated mTOR in its downstream targets P70S6. Kinase and 4-RBP1 were increased in response to leucine, and this response was greater at 30 minutes as compared to 60 minutes. It also means that L-leucine increased signaling components responsible for increased protein synthesis in murine myoblasts. Of the two major upstream regulators of the canonical AKT, mTOR, P70, S6, kinase pathway, myostatin, and IGF-1, neither expression were altered despite downstream target responses. What this further means. Response to leucine was rapid in action and lasted less than 60 minutes. This is of critical and biological importance because it is impossible to overcome anabolic resistance during disease states and aging by altering the mTOR response and its downstream targets by L-leucine. The mechanism by which leucine regulates these signaling pathways is not known. Our earlier experiments suggest that leucine may decrease autographic flux because the LC3-2 was not expressed in response to leucine. Further directions to go. Leucine can be used as a supplement in aging and patients with sarcopenia. The signaling response to leucine provides evidence of leucine as a narcocortical agent. The exact molecular mechanism by which leucine overcomes mTOR resistance is not known, and that needs to be studied. Thank you, Dr. D, for teaching me so much about skeletal muscle and leucine, and Gia for teaching me everything there is to know about the lab. Thank you to everyone in the pathobiology department for being so kind and helpful.